Hello, everybody, and welcome to another TN2 seminar. Today, we have this lunch seminar dedicated to brain imaging with some really interesting speakers. So these are some general introductory remarks on Zoom. As I'm sure all of you are aware, this is a webinar. So the webinar in Zoom means that you cannot see each other. You can only see us in the panel. And if you want, you can change the screen, so you can increase the size of the presenter or the slides. You can change that however you want. And if you want to ask questions, please do so in the Q&A tab, not in the chat. And we will monitor the Q&A for questions and then answer them as we move along with the speakers. So something about TN2, if you have not seen one of our webinars before, we do these regularly. And the aim of TN2 is to translate between fundamental and clinical neuroscience. That's why it's called TN2, Translational Neuroscience Network. And what does this mean? That means that we usually have talks on a specific topics, like today on brain imaging, one with a more fundamental character and the other perhaps a more clinical or more closer to industry or their patient. And then we will discuss how these two different subfields influence each other. Our meetings have now moved, of course, to Zoom because of COVID, but because we're almost coming out of COVID, hopefully, also organizing a regular conference in person, and more information on that will follow. And the focus is usually on neurology or psychiatry or a combination of both. And these lunch webinars will continue until June. So please have a look at the website and the schedule, and hope to see you again in the next one after this. So today we are focusing on brain imaging. That's one of the research pillars in Amsterdam neuroscience, as you can see here. And within brain imaging, one of the leaders is Professor Liesbeth Reyneman, who is a neuroradiologist at Location UMC, who will now talk to you a little bit more about the research pillar as a whole.
Yes, thank you, Menno. So the brain imaging program, besides providing an excellent research infrastructure for the Amsterdam neuroscience community by optimizing available scanners and analysis platforms, focuses specifically on fully translational from bench to bedside via micro EEG, PET, SPECT, and MR studies, a model focus, as well as minimally invasive imaging technologies suitable for personalized medicine and drug targeting strategies. So I think the two speakers we invited today perfectly match that profile. Natalia Petridou is from the high field group of the Department of Radiology at the University of UMC, and Henk-Jan Mutsaerts of the Department of Radiology here at Amsterdam UMC. And together they nicely cover these three aspects: the translational, multimodal, and minimally invasive aspects of our brain imaging program, since they will be talking about non-invasive assessment of brain hemodynamics. Natalia will be talking on the BOLT technique and Henk-Jan on the arterial spin labeling, or ASL. In that sense, multimodal and definitely minimally invasive. There's also a translational potential, since Natalia's talk will be on a micro or meso level and Henk-Jan's more on a macro clinical level. And we would like to discuss these aspects in the closing remarks. So each talker has 20 minutes, and then we have in that session a 10-minute discussion, and then followed by the second speaker, and then the final remarks, of course. So herewith, I would like to introduce Natalia Betridu. As I said, she is in the high field group at the Department of Radiology at the University Medical Center in Utrecht. She's a biomedical engineer by training and has a minor in neuroscience from the George Washington University in the United States, and somehow ended up here in the Netherlands and conducted a postdoctoral research fellow before that in Nottingham. She studies the relationship between neurons and vessels in the brain for advancing fMRI measurements. I think very exciting topic, and I'm very happy that she's willing to provide a lecture on this interesting field. Please, Natalia, the floor is yours.
Thank you, Lisbeth. Thank you for the invitation and the introduction. Let me share my screen with you. Okay, so I trust that you see my screen. All right. So the title of my talk today, like you saw, is Vascular and Neuronal Contributions to Bold fMRI. And the reason this project generally exists is, or is performed, is twofold. First is more on the neuroscience context. So we want to understand and quantify what information we can extract from bold imaging. And the other part is we want to characterize and model properties of the vasculature and hemodynamics from intracortical vessels in the brain. And that is not only relevant for quantifying bold but in the neuroscience context, but also in a clinical context. For example, by understanding or diagnosing small vessel diseases. Specifically, what I'm going to talk about today is the specificity of T2 star weighted bold at 7T. That is the usual gradient echo fMRI that we perform, the most commonly used. And I will address the specificity with respect to the intracortical organization of the vessels and with respect to neuronal activity as we measure with intracranial electrophysiology in human participants. So before I get into the gist of it, I just wanted to highlight the advantage or the beauty of 7T as compared to 3T. And this is an example that I have of, let's say, conventional fMRI you may perform at 3T. So that would be something like 3 by 3 by 3 millimeters isotropic voxels. And you see that on the left side. And I want to compare it with state-of-the-art 7T fMRI that we do these days, which is at 0.55 isotropic, roughly. So basically, we have a difference in resolution of a factor of more than 160. And I want to highlight here that the underlay that you see is the actual fMRI image in both cases. And the color overlay is the bold activity that we measure. And I think you can appreciate very nicely the difference in size and the difference in detail we can get at 7T. Now, what do we measure with such small voxels in the brain? Now, if we zoom in a small part of gray matter, this is what we have in there. This is a picture of the vasculature. It's a vascular corrosion cast ex vivo after the tissue has been washed away. And I want you to notice the scale bar here. And then you can appreciate what amount of detail and diversity we can get in each one of those voxels that we measure. So what happens when we have activity in the brain? Here we have, this is a surface of gray matter, this is a white matter, biosurface, and here we have our cortical layers and the majority of the capillary beds or the microvessels. So when we have activity, it's initiated somewhere within gray matter that sends a signal to the vessels to send more blood in. So it comes from the surface, blood diffuses in the capillary bed, oxygen diffuses to tissue, and then deoxygenated blood drains back towards the biosurface. This is what we actually measure with bold. Of course, this happens because we have neuronal activity, and this is also a very complicated organization within the cortex. We have several cortical layers with different functions. And this organization, the neuron organization, is not necessarily matching with the vascular organization. But this is a driving force of a signal. So one of the important parameters to know to understand how precise our bold can be to measure this neuronal activity is a point spread function. So basically the spatial specificity of the bold signal. And this is what we investigated in this case for anatomically and functionally defined column-like structures in the cortex. So this is what you see on the left side. It's in the visual cortex, visual area B2. You see the brain here, an inflated brain, Actually, my cursor disappeared. There we go. I'm zooming in, and the color overlay is the bold activity. We're looking at this structure here. It's because it's inflated, those structures look like stripes, closer to white matter on the left, closer to CSF on the right. And we can quantify the point spread function of the bold given those structures, and also their anatomical size, which we measure with T1 weighted imaging. This is what you see on the graph on the right. White matter would be here, CSF would be on the pile surface would be on the right side. The anatomical size generally doesn't vary across cortical depth, and we can compare that with the bold point spread function, which is actually pretty close, is less than half a millimeter within the deeper parts of the cortex, and it's increasing towards the pile surface. And that relates with the size of the bigger vessels that drain blood towards the pile surface. Of course, the amplitude and the point spread function relates to the different vascular compartments, as I already alluded. And we see this in a bit more detail. So this is a very high resolution fMRI. You can start differentiating a little bit the different vessels in the cortex. Let me run you through the slide. So we're looking again at visual cortex. This is a zoomed view of primary visual cortex. This is the T1 weighted scan that I was talking about earlier. And with such a scan, we can also visualize layer 4, striae of Gennari in this case. In the middle, I'm zooming in further in primary visual cortex. This arrow, the red arrow, points to the layer 4, the striae of Gennari. These views are actual fMRI, and because the resolution is so high, we can start seeing the layer 4 as well, a little bit. And here we have our activity overlay. This is a task that's very basic, feed forward from the eye, majority activation in layer 4, and we can nicely observe that we have the majority of activity in this illustration, indeed, in layer 4. I'm showing it with my arrow. But at the same time, we can also observe other parts of activity which relate to intracortical vessels. They're sort of orthogonal to this view. And also we have pile veins that pop up. They're also marked there. If we take profiles across cortical depth, again, you're looking at a similar view as before. White matter would be here. Pile surface would be on this side. We can get different kinds of profiles. So if we only include, let's say, an area like this, we would get a bump in the signal. This is our bold amplitude, what you're looking at. A bump in the signal around layer 4, and the more of the intracortical veins we include, or the pile veins we include, then we do see a linear trend of the bold amplitude toward the pile surface. So a different kind of behavior of the signal, depending on the vascular composition you have in your measurement. And what we found is that the majority of the signal, this trend actually relates to the baseline blood volume, the blood volume capacity of the vessels that you're measuring from at baseline. Again, let me run you through the slide. We're still in visual cortex. This is a very colorful view that highlights how we sort of organize our profiles in cortical depth. So we go from deep cortical depth, middle, and top. This is the color scale that you see in this slide. And we can also examine the pile veins separately. You see an extraction of the pile veins here at the bottom left, which correspond to this, the same data basically. And then we look at our estimate of the baseline venous blood volume. And we can break that down into all vessels. That would be from our gradient echo fMRI. And we can also examine what microvessels are doing independently. And we measure that with spin echo, which is a microvascular weighted fMRI. And clearly you see this linear trend. The blood volume increases with respect to the vessel size, while for microvessels, this is pretty much flat across cortical depth. If we would normalize our bold amplitude with this baseline blood volume would actually remove this bias towards the biosurface. And that would give us an advantage to be able to extract microvascular responses or features within gray matter. This is what I'm showing here for one example. Basically, now you see something similar to before, but now it's bold signal change, all vessels, microvessels. I will be referring to that like this. So grain and echo and spin echo. And then if we normalize the signals by the venous blood volume at baseline, then we get similar profiles, basically. This is what I want to highlight here, that and that looks pretty much the same. Now, we can also extract different features from our measurements that may be generally interesting to know, to understand the bold signal, but they may also be very useful as markers to perhaps characterize possible problems with vascular function. One of them may be the ability of vessels to dilate or cerebrovascular reactivity. And we saw here that it's actually quite a big difference between microvessels and larger vessels in the cortex, in the sense that microvessels have a much lower capacity to dilate as compared to larger vessels. At the same time, the ability of vessels to carry oxygen or oxygen saturation is pretty much the same, irrespective of vessel size. And this is what you're looking at here in this graph. That can be estimated with a so-called M value, which is from calibrated bold. So one could say that sometimes we don't get a very strong signal from microvessels in our measurements because they don't have a very large capacity to dilate, potentially. And another interesting part to look at is the temporal dynamics of our bold responses within gray matter. And this is an earlier study that we did where we identified that the early part of the response, so basically I'm talking about the beginning of it from onset to peak, seems to be microvascular weighted. So again, here I have this illustration of the vessels within the cortex broken down by schematically at different cortical depths. And those are the curves that you obtain from such a segmentation. The large one is the really big bile veins, so that's clearly different than everything else. But within gray matter, we see a difference in amplitude, which we're also seeing in the profile. So it's a lower amplitude, deeper layers as compared to the higher layers. And here, this purple curve is a microvascular response. And like I said a moment ago, if you look at the beginning of these curves, they're pretty much the same. So up to the time to peak, the T2 star weighted bold seems to be microvascular weighted. Now, all those things are really interesting to know and be able to characterize because they may actually be relevant as a marker of disease or a marker of disease progression. And this is a slide by my collaborator, Thijs van Os from the LUMC, where they have observed that both the amplitude and the timing of responses may differ between healthy controls and patients, in this case, cerebral amyloid angiopathy, difficult word, patients. So this is something that we're going to look at with Thijs in a project that we just started. Now for the second part, what about specificity to neuronal activity? So this is something that we looked at by comparing bold measurements with intracranial electrophysiology or ECOG. This is what I'm illustrating here in this slide. We have a rendered brain. The color overlay is the bold that we measure from sensory motor cortex. It's a hand movement or finger movement. And the dots that you see on top of the brain are electrodes that are implanted on top of the cortical surface. And the signal that we measure from such electrodes is quite rich. This is an illustration of a power spectrum of such a signal. So the x-axis in this case is frequency. And for everything that I'm going to discuss, we're going to zoom in this high frequency broadband power between 65 and 95 hertz. because it has been shown to be closely related to neuronal population spiking. So one interesting question, of course, to look at is what about the spatial match? This is what I'm showing here. For a high density or very small grid who had its sensory motor cortex, zoomed in here in this middle panel. The dots are electrodes, like I mentioned a moment ago. We split sensory motor into S1, somewhat sensory, and M1. On this side, the pink part is the hand knob. And here, what I'm showing is a contour plot from this grid of the activity patterns obtained from the ECOG in the HFP and 17-volt. The task was a movement of one finger. And visually, you can already appreciate that both the location and the spatial extent of activity is actually very, very close. It's about a 70% match in this illustration. This excluding the large spiral veins. If we include them, then this pattern for the ball becomes a little bit more blurred, but of course the location is still the same. And generally, the take-home message from this slide is that the hotspots of activity are actually extremely close. So it's a very good match that we get. And the difference that may be illustrated here, so it's the foci for ECOG and BOLD, could also be due to our measurement error, basically, or in aligning the grid on top of the BOLD maps. And here it's the hotspots for moving 3 different fingers. Now we can look at the spatial match within gray matter, going back to now laminar profiles. But now a slightly different example, and I want to show you measurements of neuron population receptive fields. Receptive fields in visual cortex, I'm illustrating here. So what this means is basically the part of the visual field, the region in the visual field that gives a specific kind of activity pattern in visual cortex in the confined neuronal population. So if we look at that across cortical depth with 70 bold, this is the left graph, white matter here, pile surface here, it looks like this. So basically the neuronal population receptive fields are smaller where the input comes in layer 4, and we have larger ones in infra and supragranular layers, which may be expected as information gets processing becomes more advanced. This is what has been observed as well with laminar recordings in monkeys, electrophysiology recordings, and we're comparing here with the red curves that you see in this graph. So it's actually very, very similar. So we can extract a lot of detailed information about neuronal activity with laminar recordings and PRF modeling. Another very important thing to look at is the amplitude correspondence. And I want to highlight here the issue of linearity or non-linearity. And what does this mean? When we analyze our fMRI data, we always make some sort of assumption, a linearity assumption, that the stimulus intensity of our task translates linearly to neuronal activity, and that translates linearly to bold amplitude. Now, normally we don't have a measurement of neuronal activity. And very often what has been observed is that there is no linear translation from stimulus intensity to bold amplitude, for example, with fast stimulation. And this is, the schematic illustrates this for increasing rate of movement of the hand. And if you assume linearity, the conventional GLM linear model with linear model of your hemodynamic response, you would get the red curve. But what is often measured, like I mentioned, is this blue curve, which appears to saturate our faster movement rates. So a question that we asked is whether this nonlinear behavior may actually relate to neuronal nonlinearity, and how would the bold relate to that? And what we found in our experiments is that the bold amplitude actually follows neuronal non-linearities, at least for the experiments that we did. Again, we're back in sensory motor cortex with our high density grids here. Oh, lost my cursor. And looking at the graph on the right, we have relative bold amplitude and movement rate on the x-axis. And if we were to analyze our data, assuming this linearity, we would get this line. However, if we're using our ECOG signal to predict our bold, we get the black data points. And comparing that with this model, comparing that with the actual measure response, they're very, very close. We do see this saturation occurring, starting from the neuronal activity part. And nicely, we can actually observe a similar thing at 3T and also in different cortices, in this case, a visual cortex. And the experiment here was increasing the rate, the flicker rate of a visual stimulus. And again, we see something similar happening. So the linear prediction is here. Here's the data with the saturation. So the ECOG based and the actual observed bold seem to match quite well. And that brings me to my last slide. What about negative bold or suppression, which is also a difficult signal to interpret? And because it has sometimes been related to hemodynamic artifacts as opposed to neuronal activity, excuse me. So we looked at that with a solid paradigm, if you will, to examine both excitation and suppression of activity in visual cortex. And we did that with just a simple feed forward from the eye to the visual cortex, and then you get positive activity, which you see here. And the electrodes, again, are the dots. And by changing the hemifield of the visual stimulus, you can actually suppress that hemisphere. And this is what is illustrated at the bottom. And we'll have in the same locations negative bold. And the graph shows you the correspondence of the amplitude of the two signals. So the red part is in the excitation condition. Each dot is an electrode. So we have positive bold with increase in broadband power. And when we suppress activity, we have a silence of broadband and corresponding negative bold. And I think I may be out of time, and actually this is the end of my presentation. And I would like to thank everyone that contributed to the work that I showed you today. And also thank the funders that made this work possible, and open the floor for questions. Thank you very much for your attention.
Thank you very much, Dr. Petridou. What we're going to do now is take some questions, and we'll alternate between myself and Lisbeth. So we have a few in the chat. I'll take the first one from Linda Dow. She says, wonderful talk. Can you reflect on whether these associations between neuronal activity and bold also generalize outside the visual or motor cortices?
That's something that we'll have to look at more carefully. So I don't want to say yes nor no. Truth is, we have not performed those experiments. I would expect that they generalize to a certain degree, but perhaps not everything. And one of the things that may be, we don't exactly know what neuronal activity is doing. So it's hard to understand what the bold is doing if you don't measure it. So yeah, probably.
I guess it's also a difficult areas to model with specific tasks that you don't know what you're supposed to expect in terms of activity, right? Because in the visual system and motor system, it's perhaps more constrained. I don't know if that's the right word.
Well, it's different. I think information processing is different. We have a little bit of an insight with a study that was conducted with Serge and a PhD student of Serge, actually, where we looked at numerosity processing in parietal cortex. So the general principles seem to hold, but then the activity is not exactly the same. So maybe you have more input actually in your input layer, and then you have more specializations. You go to super and different granular layers, and then you would get a different profile of activity. Oh, yeah. We did see that both may follow that. So,
okay, interesting. Thank you.
Yes, we have a question from Stefanos Bouskas. Thank you for your talk regarding the bold non-linearity of signal strength. To which extent do you think we've been interpreting fMRI signals wrongly?
That's a very difficult question, actually, because it always depends on the kind of analysis. I'm not sure how to answer that. Yeah, I would return it with an advice that you have to have a critical eye on what kind of model you assume when you analyze your data. I think that's the best way to answer that. There are some more advanced models where they assume a given degree of non-linearity, and then it's okay. If you don't use such a model, maybe you actually have quite an error in the interpretation. But it's very hard to say in general terms what what it should be. Can I ask a really quick question? Should I stop sharing my screen? I'm not sure what people are seeing at the moment.
Oh, we see the thank you slide, so you're continuously thanking the team. That's okay.
That's a good thing. Okay.
There's another question by Arnoldo Castro. Thank you for the great talk. If activity in different but parallel cortical layers is synchronized, do these bold signals affect each other, and can these effects be corrected for with the new technology with higher spatial resolution?
That's a very interesting question. There's two kinds of interactions. One is the neuronal interaction, of course, because you have signals bleeding from, or neuronal communication between layers. And you also have the draining of blood. So from deeper layers, it goes towards the surface. And they're not necessarily, they're not the same. So you will have, just by draining, you will have an effect that has nothing to do with neuronal activity. And on top of that, you would have the neuronal activity. So they relate and not. You cannot correct it only by extremely high resolution. I think you need to have some additional correction there. Not I think, I know. Either that's a model like the PRF model that I showed earlier. Maybe I should go back if I can.
Yeah, you can.
I cannot apparently because it's frozen.
Oh.
Where we have this U-shaped, we actually apply the model to try to extract the neuronal component and ignore, in simple terms, the hemodynamic component, and then we get this U-shaped curve, which is very much unlike this linear trend that increases. I'm hoping that people can see my hand waving.
Yeah, we can see it. Yeah.
So you can use other methods to correct. This is one. Another one is to try to extract the hemodynamic components, the bias that you don't want, either with data or with models, which are out there, by the way.
Great, thanks.
We have another question from Stefanos Bouskas. What are the implications of bold non-linearity for measures such as functional connectivity?
This, you mean, I will ask him to elaborate a bit more, with a task or without?
I think without task.
That's a...
Yes, he says without task.
Without task. I guess, well, the truth is, I haven't done it. I can speculate. I would imagine it's the exact same thing, basically. There is no reason for neuronal activity to be different when you have a task or when you have an internalized activity. So I would expect the signal behavior to be the same. But of course, it's more difficult to know what the neuronal activity is. Again, we fall back to the same problem, that where are you? Is it, if we would take at least the example of the fast versus slow, do you have a fast activity or do you have a slow activity? At the end of the day, I think what is needed is a really good hemodynamic model where you can infer your neuronal activity with more reliability. Does that answer the question?
We'll see. I hope so. I think we are running a bit out of time. We can keep them for the general discussion at the end of the next lecture. Maybe it's a good moment to introduce our second speaker. I hope everyone will stay and listen to the talk by Henk-Jan Mutsaerts. He's an assistant professor here at the Amsterdam University Medical Center. Sorry, I have to think about it a little bit. But at the location UMC, he was originally trained as a medical doctor, actually never pursued a career in the medical field so far, following his PhD work with Aart Nederveen and I think also Thijs van Os on arterial spin labeling. We actually invited Henk-Jan for a talk on the blood-brain barrier with arterial spin labeling, but I think that work is still a little bit too early, maybe another TN2 talk on that. He'll touch base on it, but mainly we'll talk about ASL perfusion as a biomarker for cognitive decline. Henk-Jan, the floor is yours. Thank you very much for giving a talk.
Thanks very much, Lisbeth, for introducing me. Can you all hear me and see me? Right, well, thanks everyone for inviting me and for attending this lecture. There's several of you who actually know much more that I saw of arterial spin labeling from the physics side that I saw in the attendees list. So I apologize for those. I'll keep it very clinically and physiologically oriented, and I'll simplify some of the technique because I'll show you some of how the technique works for those who see your ASL for the first time. And I'll take you through some of the research that we are doing. And thanks, Lisbeth, for introducing me, so I don't have to repeat that. What I would like you to think about during this talk as well, if you doze off, what does it do with your perfusion? If you take a cup of coffee, because this is a lunch seminar, and you eat some lunch, what does it do with your perfusion? Think of that, the blood, you have a blood supply and you have a blood flow demand. How does that differ when you have a cup of coffee and when you're having a lunch? And I'll come back on that later in the context of developing ASL perfusion MRI as a cerebrovascular age biomarker for cognitive decline. And we'll come back at cerebrovascular age. What is aging? Well, if you have these two patients coming in your outpatient clinic, polyclinic, then you'll immediately have a different idea of what their chances are for disease or for death. And you could think of aging as a sort of accumulation of wear and tear. And accelerated aging could therefore be a sort of indicator for disease or probability even for death. And that's the idea of comparing the biological age with the chronological age. As a chronological age we have from the clinical data, but if we then make a scan, in normal volunteers, we will assume that their biological age is the same as chronological age. But if somebody has a smoking or sedentary lifestyle, then maybe the biological age, and we're looking at the cerebrovascular age, looks higher from the biomarker perspective than if you look at the chronological age. And I'll come back to that later once I've explained ASL. But that's the idea of biological versus chronological age. Nothing new about that, but we are going to apply that on ASL. And why do we want an imaging biomarker in the first place? Again, zooming out from the very nice, more detailed presentation of Natalia. Well, we obviously want to be able to, in an earlier stage, diagnose diseases, the type of disease. Maybe you want to predict also to patients that have some form of genetic or familial predisposition to get a disease, to take away some of their fears. But also, last but not least, if we can actually do something about diseases, and a lot of cognitive decline, actually the best things we can do is try to change lifestyle and behavior, is to actually monitor what happens in the brain. And for that, it's good to recap the so-called Clifford-Jack diagram, in which you, once the patient comes in your outpatient clinic with cognitive deficits and you do an MMSE test, then you're probably too late. But we are interested in biomarkers that very early could predict us who is at risk of cognitive decline. And if you then sort of flip this diagram 90 degrees, then one thing that is very interesting is that we know for a long time already that blood pressure, as one of the cardiovascular risk factors in midlife, is associated with cognitive decline later on age. And we do have a sort of more looking at the hardware of the brain rather than the software, the cognitive test. We do have some biomarkers that we can actually use, and it's been used for looking at brain age. But those that you see here, so volume loss, atrophy, and white matter lesions, sort of vascular structural changes, those are rather static. So they accumulate over time, and they are hardly reversible. So the idea for ASL to fit in this is that you have something that is a dynamic, physiological, hemodynamic biomarker. And so maybe that is something that will change earlier in the disease, while it's still reversible once we have something to do, something about it. And the side effect of something that is very, sensitive is obviously that it will also be sensitive to physiological variation, like the cup of coffee in the lunch, and I'll come back to that later. So how does ASL work? Here you see a typical MRI layout. And here you see it in a very layman the picture. So in the end, we want to have a scan that gives us a proportional image to how many mils of blood flow through 100 grams of brain tissue each minute. And as you see here, that's about 3 times as high in the gray matter, where the action happens, as we learned from Natalia, than in the white matter that just needs some blood flow to stay alive and to transfer the signals. And then if you look closely, there's even contrast between the white matter and the CSF, depending on the SNR of your image, which we'll see later. What we're doing is in the arteries, well, we're labeling here in the proximal plane, but the arteries are the only thing that come up. We're labeling spins, and spins is MRI jargon for water protons, so H pluses that are abundant in water and thus in the human body. And that's why it's called arterial spin. And the labeling stands for the inversion of the magnetization. So for the non-physicists under you, just forget it. What it means is that we are changing the magnetization in a plane proximal to the tissue where we want to, distally, where we want to measure perfusion. So we allow for a post-labeling delay. That's about 1.5 to 2 seconds typically for the blood to arrive in the brain where we want to measure perfusion. And so this is around 1.5 to 2 seconds labeling, 1.5 to 2 seconds PLD, post-labeling delay, and then the readout. And if you ever get the chance, stay next to the scanner, because what you'll hear is beep, beep. And that's really one of the main side effects from ASL. It's super noisy, and the pause here, the PLD, makes it very annoying to the patient. So that's the only real disadvantage. What should it look like for a single subject that you've seen before, or averaging in MRI? For a population average is that going back, the brain weighs approximately a little bit less than 1.5 kilos, the size of 2 hands, and we have about 750 mils per minute flowing in, and that averages to 50 mils per 100 gram per minute over the brain. And again, a little bit more for the gray matter than for the white matter, if we have the resolution to measure that, as I explained to you before. And that's what you see in this contrast. And one reason why it's not always looking as it should is that the timing is very essential. And this can give you a so-called vascular artifact, which to me is very interesting. I'll come back to that later. So if you are too early, you can assume that the blood is still there, so you haven't measured any labeled blood. And then a little bit later, you see the labeled blood coming into the major arteries, the ACA, the MCA, and later in the posterior territory. And if you wait long enough, you'll see that perfusion is that the blood is sort of distributing in the tissue where you measure how much perfusion we have. If we wait too long, the blood, the labeled blood signal will decay. That's actually one of the also disadvantages of ASL that I will show you later. ASL has a relatively low SNR, and that's also one of the reasons that it took quite long to be invested in clinical workup. One of the reasons is that this labeling is never 100% efficient, and that efficiency can vary a lot with how you position this labeling plane, and you can probably appreciate that this is more tricky with the sort of tortuous posterior circulation than with the rather straight anterior carotid arteries. And those arteries becoming more tortuous with age, you can see that this actually differs. And if you see this patient that walks through in and out your outpatient clinic, that it's not a hemiplegia, but it's just a labeling asymmetry. The second is that the blood decays quite fast, even quite some faster than water PET, which is the ground sort of golden truth of perfusion. So that the blood T1 decay is in the same order as it takes for the blood to arrive in the brain. So on 3 Tesla, that means that half your signal is gone in about 1.7 seconds, and that is dependent on hematocrit. So if you don't account for hematocrit, we might see disease control differences that are not realistic. And then the third is obviously that the amount of water or protons in the brain is only a few percentage for blood versus all the other protons or water in the tissue. And the cerebral blood volume is relatively small. That means that we need to lower the spatial resolution, much lower than what Natalia has just said, typically 4 by 4 by 4 millimeters. And we have to carefully time. And one assumption is that the blood never leaves to the veins with some signal left, unless you have what you see here, a child with sickle cell disease where the flow is much faster, very low hematocrit, so their blood T1 stays much longer. So these are some examples where knowledge of physiology clinics are very important in the interpretation of these images. So going to our research from the explanation of how ASL works, and again, the bare basics, we have 3 pillars that we find are essential to develop this technique. One is the standardization. We need to speak the same language. We need to be able to interpret what we're seeing, and I will focus more on this later. And we want to apply it in different diseases and in commercial activities. And we have a lot of nice community activities. So one interesting thing is BITS, brain imaging data structure, to have the same language of how we save our data for research. And we've developed that for ASL. We have a standardized pipeline where we actually also put a lot of effort in just the structural processing, because that's your reference for where we are measuring perfusion. And this pipeline is used in a lot of regions in the world. And I'm, of course, cheating here, because if it's even 1 or 2 cities in the States, then the whole country gets blue. But there's a lot of users worldwide, and which we also see back in the community meetings. So it's nicely growing. We do a lot of QC. This is also nice. There's this Philips IntelliSpace design discovery. And we are trying to make Explore ASL incorporated in this software, so radiologists can actually use it in departments and hospitals that are not academically, that are not specialized to use new, novel, cool MRI techniques. And we have a Eurostars grant where we want to make ASL ready for clinical trials. So to really standardize the processing, make it easy to upload it, and to have actually a flow phantom that is MRI compatible, where we can actually measure what the differences are between MRI scanners, because MRI scanners are used to take pictures. They're not essentially measurement devices because they give arbitrary units. So we have a phantom to really calibrate that. So a lot of cool research going on, but now I will focus on cerebrovascular age. Before we go to a vascular component, I will skip the structural component. These are sort of the main three components that we like when we interpret the ASL signal. I will go to the functional component. As Natalia said, the idea for ASL was always from the neurovascular coupling. And so we try to measure something because a disease hits the brain in regions where there will be less function, and by that, less use of glucose, less use of oxygen. So here you can see on the FDG PET, and perfusion would give you similar patterns because you need blood flow. And so this is about the blood flow demand. This is where ASL can help a lot. But what we've seen, which I will show you, is that there is this timing caveat. So I will skip this. Well, this is actually another good example. If I just did a t-test for genetic frontotemporal dementia to compare pre-symptomatic carriers with non-carriers, with a simple t-test, there was too much physiological variability, also in the arrival of the blood. So I didn't find a correlation. If I look at an interaction with age, then you see that those that are carriers of this genetic disease, they actually go down in their perfusion as they reach the age of onset. And you see that that's significant in the so-called signature regions of FTD. So definitely ASL has a place in the demand of blood flow, so the neurovascular coupling, but there is this very prone vascular artifact that I'll come back later that makes it quite difficult. So again, the physiological variability. I'm now around halfway through my presentation. This is a PhD student that I supervised in Belgium. There's a lot of, she studied about 500 papers from different modalities, and there's a huge amount of perfusion confounders, and that leads to, now ASL has developed better to the same variability between weeks as water PET. But it's essential now that the technique SNR-wise is sufficient, that we focus on how do we cope with caffeine, meal planning, and other reasons for physiological variability. So to answer your question, caffeine does increase neuronal activity, but it will constrict your vessels, so your perfusion actually goes down. And there's a oxygen extraction fraction buffer for that. And with a meal, there's CO2 coming up from the metabolism that will actually open your blood vessels. So the question then is, do we create a normality atlas for some of these main perfusion confounders, or do we create standardized operating procedures where you ask the participant, did he drink coffee, did she take a meal, or that you ask them to come in sober? And these are questions that we're still trying to solve. Going to the, I have to look at my time. Going to the vascular component, looking back, this is how we want ASL to behave, but this is how it behaves in reality. In ADNI, for example, had a lot of ASL scans that were in a quite early phase imaged. So if you have patients that have, for some reason, proximal delays or distal resistance, the blood just doesn't flow as fast as it does with somebody who's young, hasn't smoked, has led an active lifestyle. Then what you see is that all the blood is still in the proximal arteries. And if you do your typical QC, then what people would say, well, the ADNI ASL is really unusable. There's a lot of these cases where the blood is still in the proximal arteries, not in the distal tissue. So we cannot measure perfusion. So to say something about the demand of blood flow with these scans is simply not possible. But what about that this actually is some signal in there that is actually clinically relevant if a whole brain cerebrovascular health is a sort of different with one patient to another. So what we did instead is use this signal. Look at, instead of the mean perfusion over a region, look at the standard deviation normalized by the mean. which we call the spatial coefficient of variance within the gray matter. And let's see how that works. And this was a first tryout with asymptomatic unilateral carotid occlusion. So the patients should have reliable perfusion because they don't have any symptoms. And what you see, if you look at this image and look at this movie again, is that through collateralization, it takes longer for the blood to reach here. And the mean perfusion is the same, or the mean ASL signal is the same at both sides, but the heterogeneity is much larger at this side. And the spatial COV did predict much better the site of the asymptomatic unilateral occlusion. But does it actually help us with cognitive decline, vascular dementia, the vascular contribution to Alzheimer? And yes, there's a lot of studies that we did where we saw that it correlated better than average perfusion with age, sex, white matter lesion load, cortical microinfarcts, intracranial stenosis, and other types of cognition. And in ADNI, it was very interesting. And if you even already took amyloid PET, FDG PET, so mind you, the neurovascular coupling, the demand of blood flow is already in the model, that still, if you add this variability of blood flow arrival, so the cerebrovascular health, to the model, that you still become better and significantly better in predicting who has AD, MCI, or normal controls. So this really got us enthusiastic to say, can we develop this some more and go back to the biological versus chronological age mismatch that I explained to you in the beginning? What would happen if we took from each brain region the CBF, the tissue perfusion, this vascular efficiency or spatial COV, and add it to a machine learning model? And what we do in a large population of healthy controls is we teach the machine learning model to predict the real chronological age from the ASL scan, from these image features, perfusion and vascular efficiency, across a healthy age. Because in healthy persons, the difference between the predicted biological age from an ASL scan and chronological age should be minimized. And that's what Matthijs Dijsselhoff, PhD student, is doing right now. And then later, we can actually use this to see if you have a patient coming in that has been smoking all his life, a sedentary lifestyle, does his ASL scan actually look older? Does he have an accelerated cerebrovascular age? And does it actually help us studying the link between the cardiovascular risk factors midlife and late life cognitive decline? So Matthijs has been...
you, Henk-Jan, to kind of wrap up.
Yes, I will do that. So we have been looking at one study this far, and I will just show you this slide, that if we only look at ASL cerebrovascular age, that there is a huge variability, so maybe physiological variability, but also flare, so white matter lesions only still don't, in healthy controls, predict the chronological age very well. But if you take structural cerebral age, brain age, and you combine it with flare and ASL, you get significantly better at predicting the healthy age. And I'm very enthusiastic to see how this will work in disease later on. So thanks for your time. Let me know if you have some questions.
Yes, thank you, Henk-Jan. I saw you were already nearly at your end, so
yeah, sorry for that.
Well done. Yes, are there any questions? Do I see any questions? Not yet. Hey. To make a link to the prior talk, is there any work on how do you see functional imaging with ASL, like typical fMRI work using arterial spin labeling? What is your vision on that?
Yeah, good question. I think a combination of both measures a venous signal and an ASL arterial signal would be nice, but and there are techniques that try to do both intermittently, so that you take two echoes, the first you use for ASL and a later echo for fMRI Bolt, and Thijs van Os has shown that it actually doesn't decrease ASL SNR too much. The tricky thing is that ASL requires a lot of SNR and has this labeling duration and PLD that is quite long. So your temporal resolution with ASL is quite low. So the things that Natalia does really it takes us another 10 years to do the same with ASL, especially at 70. But there are certainly ways to combine it.
I can also ask a question. I was just wondering, there was a lot of hype a few years ago that perhaps ASL is ready to replace perfusion PET, and with that maybe save quite a bit of money because PET is so expensive. So do you think with these recent improvements in your pipelines and so on, are you ready to let go of perfusion PET? For clinic and research, I guess. There's maybe a different question for both.
Maybe it's nice to start with the joke that I was in Toronto at the AAIC with my nice GenFi poster. And there was somebody coming, you are using the poor man's PET because ASL was sort of considered this poor man's PET. I think the main thing now is this physiological variability and interesting to talk with Bart what he thinks about that from the perfusion PET perspective. The main hindrance still is the timing. And so with ASL, we are getting better at correcting the timing, but that's the main hindrance. And again, also a source of signal that PET doesn't show this vascular delay. So that's why I tried to zoom in it from a positive side, but still, even with the newest techniques, this delay is the major problem. So if you have younger brain tumor patients, then ASL would be perfectly able to, if I may say so, Bart, replace water PET. But especially if you have older and more vascularly prone patients, then still we have to see if we can actually get the same reliability in patients as water PET.
And I guess that also means that because of the delay, that I understand the delay gets worse if there's vascular pathology, but does that also mean you cannot really decrease scan time because you always have this physiological component that you, of course, cannot accelerate? Or do you think there will be technological advances that will make it quicker to scan?
Yeah, there are certainly technological advances, and we were trying some of them out right now. Also to scan the blood-brain barrier, and I didn't talk about it now, but that's the point. Do you want to accelerate it, or do you want to use that gain in time to actually measure other very interesting metrics? So yes, there is a possibility, but still most of those, as you can appreciate, are developed and tested in healthy young volunteers. So there is a huge possibility for us to try it in patients.
Lisbeth, there's also a question in the normal chat, I see.
Yeah, from Niels de Jode. Thank you for your presentation. Regarding the ASL phantom you mentioned, are the results that come out of it actually used to correct the data, or do they serve as a control to see how reliable the scanner at a specific site is over time?
You could do both, but at this time, mainly because we have to calibrate the calibration phantom still. That's also a work of research as we go along. It's rather the second. It's more realistic that you, for example, we have such a phantom that we would scan it, and I have to look at Joost if we can actually afford that. But every morning and every evening or every week, you scan the phantom and you take the temporal fluctuations into account, and you do that at multiple sites with different scanners. I don't think it's realistic to do that for each patient and really patient-wise sort of correct for
that. There's another question in the chat I see by Natalia, actually. It sort of links to what we were discussing before, the link with PET, that ASL has a lot of physiological variability that you have to know in order to get correct measurements. But is that also currently the same in PET? Do you know?
I would say so. I have to discuss it with Bart. I'm mentioning Bart because I saw he was on the participant list. Yeah, it should. And this is what we also measured when we compared at the time with the Philips scanner at the AMC to the EPI ASL. even an older ASL technique, comparing it with PET between weeks, maybe not between minutes, but between weeks, you get similar variability, which must be dominated by physiological variability.
So I guess then the difference would be how you, the quantification potential, not the physiological variability, right?
Yeah, so this arrival, given that this arrival time or this delay time vulnerability of ASL, that would add additional variability. But again, we did the study in young, healthy volunteers, so we didn't have that problem with nice techniques. So yes, if you, again, older and more vascular diseased, then PET will probably win in terms of physiological variability, I would predict.
I think this is a nice moment to end the last session of the last speaker. Thank you so much, Henk-Jan. And I think we can continue now to a more general discussion with both speakers. Well, we already had a talk on a discussion on the pros and cons of BOLD versus ASL, and maybe there's already one question here in the chat. Shall I pose it?
Yeah, sure.
I know.
Oh, and by the way, the other questions for Natalia, we couldn't answer all of them before. Natalia answered them in the Q&A, so you can also have a look now at the tabs for answers. But Lisbeth, go ahead. Yeah,
I'll just address the last question since I know this is recently posed. Could we discuss or speculate, this is by Arnaldo Castro, could we discuss or speculate on the difference in saturation rate and non-linearity of BOLD signal in relation to stimuli strength between old and young participants and between healthy participants and participants with neurovascular conditions, for example?
I'm sure there's a lot of literature there that I'm not aware of, so I will have qualify my answer there. Well, you would, first of all, we know there is a reduced vascular reactivity with age. So perhaps you could expect, assuming that neuronal activity sign is the same, you could expect that you have accumulating non-linearities from the vasculature. But of course, there's also, I'm sort of thinking out loud right now, there's also a considerable buffer that the vasculature has. So perhaps in healthy conditions, you wouldn't notice such an effect, but if you have some disease, I would expect that you would. So if you have plaque accumulation or the vessel wall is for some reason impaired, then I don't think I don't know that yet, at least, but I don't think you can assume that there is a one-on-one relationship anymore. Hopefully we'll find out in this project that we started now.
Natalia, you measured it with healthy volunteers, I guess.
Yeah, well, first we need to know that, indeed. But also with, we want to see what happens in patients with CAA. So this is the project we started now with TIAS, and at least that's a very nice model because it's a very, very well-defined disease of the vasculature.
Yeah, yeah, that's very interesting.
I was also thinking, I did some ASL work a long time ago, and what I also noticed then in the healthy control sample that there's a large impact of sex on all the amplitudes. So I was wondering, Natalia, do you also see that in your 70 BOLD work, that there's large sex effects?
Not in BOLD, but you should be able to see that in CBF. So I'm going to put back in Henk's court.
What do you think, Henk?
Well, the main thing there is, of course, the hematocrit difference between the sexes, but also the arrival time or the sensitivity to post-labeling delay, the arrival time difference, and CBF itself it's actually a good question. So we would have to, I would have to go back to the PET literature to see, because they don't have this hematocrit. Well, I mean, they don't have the hematocrit effect on blood T1, but they do still have a physiological compensation for hematocrit. And so the question is then, is if you would sort of statistically correct for the hematocrit difference, does the CBF still differ between sexes? And these are very, very interesting questions. And if that is still the same between young men and women, and between old men and women, and diseased men and women, or if there's an interaction there.
Yeah, that's, I guess, one of the main problems, especially in maybe neurology, that there's an interaction in many disorders with sex, but people haven't really studied it too much. So that I think there's an important role there for, well, us as the field, I guess.
But hematocrit also changes with respect to the vessel size, right? So wouldn't things equalize at some point the moment you hit the microvessels? I don't know. It's just a thought.
Well, I do know that it makes it more difficult because there are sequences that measure. The easiest is to sort of measure the blood T1 venously, and now there are new sequences that measure it sort of arterially. But that's in a quite large vessel because otherwise you don't have the resolution. So how that equates to the blood T1 and the hematocrit in these micro vessels, and also the differences that the red blood cells tend to, I think, what was it again? They tend to sort of go into the middle, and the edges have a lower hematocrit, and viscosity differences. That's our database group does some work with sickle cell disease because there the differences are much larger, and the effects are much larger. But it's quite interesting, but difficult to measure in vivo.
Yeah, but with a given size of a red blood cells, at some point there wouldn't be a difference between males and females. So that's only the capillary bed that you would have that, right?
Yeah. I'm afraid we are running out of time. So I would like to thank you both for your fantastic talks. And I'll briefly share my screen to let you know that there is going to be another seminar on April 21st on mood, anxiety, psychosis, stress, and sleep. That's a lot, but you can call it MAPS. I hope to see you there. Follow us on our website, on Twitter and LinkedIn. And if you have any questions, you can always contact us, and a recording will appear once processed on the website as well. So thank you again to everybody, and especially, of course, Henk-Jan and Natalia, but also Lisbeth, for joining us, and see you next time.